Hi, my name is Caitlin McLean, and this is my nerve impulse project for Bio137. I am going to start my project off by talking about synaptic transmission. It occurs at the synapse, which is where two neurons come close enough together to share chemical cells from one to the other. They are separated by a small place called the synaptic cleft. The presynaptic neuron is where the signal is initiated, and the postsynaptic neuron receives the signal. In the presynaptic neuron, there are neurotransmitters packed into small sacs called vesicles. When acted upon by an action potential, the vesicles bind to the membrane and release its contents into the synaptic cleft. From then, they will bind to the receptors and could cause an action potential to occur in the postsynaptic neuron. The remaining neurotransmitters left in the synaptic cleft will either drift away in a process called diffusion, or they will be put back into the presynaptic neuron and put back in the vesicles and can be reused and recycled. And this is their process called reuptake. Moving on to action potential. First, I'm going to show all the parts of a neuron. This is the cell body, and it is the basis of the neuron. These are dendrites, and they receive signals from other neurons so action potential can occur. The green spaces along the axon are Schwann cells, and they allow nerve impulses to travel along the axon. They are individually wrapped in a myelin sheath to allow protection so the impulse may continue. In between are the nodes of Ranvier, which allows for speeding up of the process so that it does not get stuck. Then the action potential reaches the end, and so it reaches the axon terminals that have neurotransmitters that allow to generate the impulse of an action potential. The neuron starts off at a resting memory potential at negative 70 millivolts, where nothing is happening and it is waiting to be stimulated. The potassium ions are located in the intracellular fluid, and it is negative. The sodium ions are located in the extracellular fluid, and it is positive. When the neuron reaches negative 55 millivolts, depolarization begins. This is where sodium-gated ion channels will open up and allow the sodium ions to travel into the intracellular fluid, making the inside positive. Once this is finished, the sodium-gated channels close and the potassium channels open up, and this allows the potassiums to travel into the extracellular fluid, making the outside negative. Now that the ions have switched place and the sodium ions are now located in the intracellular fluid, the action potential can travel along the axon to the axon terminals, meaning action potential has now occurred. This can happen due to external sources such as pain or pressure. After action potential, everything can move back to its original position. The sodium ions located in the intracellular fluid will move back out into the extracellular fluid. The potassium ions located in the extracellular fluid will move back into its original position in the intracellular fluid. This happens through a sodium-potassium exchange pump, which lets 3 sodium ions move out and 2 potassium ions move back in. This is called repolarization. Once everything is reset, the neuron can go back to its resting membrane potential at negative 70 millivolts. To show all of the things I used. For the dendrites, the axon terminals, I used pipe cleaners. For the Schwann cells and the nodes of Ranvier, I used felt. For the ions, for the sodium ions and the potassium ions and the nucleus, I used these craft fluff balls. This poster for the synaptic transmission I used a felt piece for the postsynaptic neuron, I used Play-Doh for the receptors, and I used googly eyes for the neurotransmitters and the synaptic cleft neurotransmitters that made it in. And then I used pipe cleaners for the vesicle board. I did felt for the Schwann cells, pipe cleaners for the axon terminals, and the dendrites. For the nucleus, I used another craft ball. For the cell body, I used a felt piece. And then I also used a light.